Now the next event that is going to take place is development of embryo. Okay, so development of embryo. Now let me just draw the structure of female gametophyte once again to make it clear for you from where this endosperm is developing and from where this embryo is developing. Okay, so this is the egg apparatus. Okay, and we have 3 antibodies. Okay, so here I told you that already zygote has formed. Okay, zygote is already formed, and then here already fused. So primary endosperm nucleus here. So earlier when we were discussing development of endosperm development started from this place. Okay, from primary endosperm nucleus. Now development of embryo takes place in this micropylar region. Okay, so in the micropylar region we have a zygote. Yes or no? Zygote is formed by the fusion of one of the male gametes with the egg cell. So we have this zygote. So from this zygote only the embryo will develop. Earlier endosperm developed from the primary endosperm nucleus. Okay, so this is the clear idea of development of embryo and endosperm. Now we are going to discuss this part. Okay, this part is already formed. Endosperm we are already done with it. Now we are going to discuss with about this embryo. Now this zygote will develop into embryo. So this zygote undergoes a division. Okay, suppose this is a zygote. I will draw a single zygote from here. So this part. Okay, so the zygote is here. So they will undergo this division. Okay, what kind of division? Transverse division, and then they will form 2 cells. Okay, so this is towards the micropylar region, and then this is far away from this micropylar region. Okay, so I'm just writing micropylar. So they will this particular zygote, they will divide and then give rise to 2 cells. Okay, so towards this micropylar region will be called as suspensor cell. Okay, and away from this micropylar is called the embryonic cell. Now this embryonic cell will divide mitotically, and then they will give rise to this proembryo. Okay, after this stage you will get a structure like this. So suspensor cell you will have here. So it will give rise to this kind of structure. So we have a proembryo here. So the cell here is called a basal cell. Okay, and these cells are called suspensor cell. And here this one is called the proembryo. Okay, proembryo. Now after this stage, this proembryo they will start developing in various planes, and then they will form a globular embryo. Okay, so this proembryo they will divide in various planes, and then they will give rise to so many cells here, and then we have a suspensor cell here and a basal cell. So suspensor cell, this is the basal cell. So this is the globular embryo. Okay, globular embryo. After that it will give rise to this globular embryo will just divide again, and then it will give rise to somewhat like this structure which look like a heart shape, and then it is called this heart shaped embryo with all these suspensor cells at the bottom and a basal cell. So after that finally we will get a mature embryo. Okay, these are suspensor cell, and then at last we will get a mature embryo like this. So this is the mature embryo. Mature embryo. Now this mature embryo, in some plants they may have 2 cotyledons also, and in some plants they may have 1 cotyledons only. Okay, so basing on the number of cotyledons, we have 2 categories of plants, that is dicote and monocote plants. So now we will discuss about the typical monocote embryo, and later on we will discuss this about this dicote embryo also. Now let us study about the typical dicote embryo. Okay, so typical dicote embryo. Now actually this development of embryo is similar during the earlier stages, okay? But as it gets mature, depending upon the plants, they develop into different kind of structure. So here we will study the typical zygote embryo. So this typical zygote embryo consists of an embryonal axis, okay? Embryonal axis. So along with all these points, let me just write down or draw the diagram also. So this is the embryo of this zygote, okay? So this whole part is called the embryonal axis, okay? So this is the embryonal axis, and then it has got two cotyledons, okay? Two cotyledons. So these are the two cotyledons, okay? Now above the level of these two cotyledons, this part here from this part, okay? This part is called the epicotyl. Okay, let me just write down this side. Epicotyl. And this epicotyl terminates into a plumule. Okay, plumule is here. P-L-U. Okay, so and below the level of this cotyledon is called the hypocotyl. Okay, so this region is called hypocotyl. And then this part is called radicle, and this radicle is covered with a root cap. Okay, so this is the typical zygote embryo. Okay, this is how the structure of this zygote embryo looks like. Okay, now next one we will discuss about the monocote embryo. Now we will study the monocote embryo. Okay, so in monocote embryo also it has got a embryonal axis. Okay, so let me just draw the diagram here. So the monocote embryo looks somewhat like this. So somewhat like this it will look like. Okay, now it has got only one cotyledon. Okay, so this is the cotyledon. Okay, embryo axis is this one. And we like above the attachment, this is the attachment of this cotyledon with this one, embryo axis. Okay. Now this cotyledon is also called as scutellum. Okay. So above the level of this scutellum is this part is called the epicotyl. Okay, epicotyl. And then this epicotyl encloses a shoot apex called the coleoptile. So we have coleoptile here, and below the region of this attachment of this scutellum with this main body, below this one, this part is called the hypocotyl. Okay, hypocotyl. And then we will have all the structures inside this one, and then we will have one more structure here. This is called radicle. This is called root cap, and this root cap is covered in a thin sheet called coleorhiza. Okay, coleorhiza. So this is how the mature monocote embryo looks like. So this is all about the development of embryo. In the next video, we will study about this development of ovule into seed and then development of ovary into fruit. So that's all for today. I hope you all understood. Thank you.